Good afternoon, children.
Good afternoon.
We'll begin with the revision test, and after that, we'll continue with the chapter cell, the unit of life. So, first, we will have a test, 10 marks test, a maximum 20 minutes, not more than that. And after 20 minutes, we'll continue with our chapter cell. So, link I will share in the chat box. If any difficulty with accessing it, let me know. Shall I? Okay, few more to join. I'll give a 1 minute for them to join. Otherwise, I will have to keep on sending the link again and again. Okay. See whether you can access. Any difficulty, let me know. So you all have submitted the revision test paper. So I'll send the marks in the evening. Now we'll continue with our chapter cell, the unit of life, and we were discussing about eukaryotic cells. Some of the basic differences between prokaryotic and eukaryotic cells we discussed, and I think we started with plasma membrane. Yes, the structure of plasma membrane or cell membrane. Cell membrane is found in all eukaryotic cells, but in certain eukaryotic cells, say for example in plants, they have additional layers too. And we learned that in plasma membrane, the basic components are proteins and lipids, and even carbohydrates are also present. And the lipids, they form the main component, and there is a bilipid layer of lipids. And how they are arranged, just like matchsticks, these are arranged in such a way that the hydrophilic end will be facing towards the center. Sorry, hydrophilic part will be facing towards the external environment or aqueous environment. The hydrophobic part or the hydrophobic tail will be within the membrane. And proteins, carbohydrates are also present in the cell membrane. And the lipid component, when we say lipid bilayer, this lipid is mainly phosphoglycerides. So lipids, different types are they coming under that, and mainly the lipids that form cell membrane consists of phosphoglycerides. Ratio of protein and lipid. So, you know that main components are proteins and lipids, but how much lipids and how much percentage is constituted by proteins, that depends on different cells. So, depending upon the cells, their ratio can be different. So, here they have given the example of human red blood cells. So, here the ratio is like this: 52% protein and 40% lipids. So, remaining by carbohydrates and other components. So, 52% of the membrane is constituted by proteins and 40% by lipids. So from that, what we understand is we cannot fix a percentage, or we cannot generalize and say that so and so percentage is proteins and so percentage is lipids. Depending upon the cells, there can be differences. And the proteins present, so you know, proteins, lipids and carbohydrates make up the plasma membrane. And the type of proteins, there are different types of proteins present. You can categorize them based on the ease of extraction, how easily you can separate it out. So based on that, you can classify them into different types. So mainly integral proteins and peripheral proteins. So it is easy to understand from this picture. Integral proteins means they will be partially or totally buried. They'll be fully embedded in the membrane. So as you can see in the figure here, just a minute. So here you can find it is fully embedded in the membrane. So this is the membrane. So it is fully embedded in the membrane. Then you can call it as integral, integrated into the membrane. Whereas peripheral proteins, so here they are not buried or they are not embedded in the entire membrane, but they are attached to the peripheral region or on the boundary or on the border. They are arranged on the periphery. So it is easy for extraction. So if you consider the ease of extraction, that is easily the extraction can be carried out for peripheral proteins, whereas integral protein, they are deeply buried within the membrane. So these are the two types of proteins. Once again, I will repeat, plasma membrane is made up of proteins, lipids, and also carbohydrates. And the proteins present can be categorized into two main types depending upon the ease of extraction, how closely they are associated or attached with the bilipid layer, and peripheral proteins and integral proteins. As the name suggests, peripheral proteins are not fully embedded. Instead, they'll be present in the peripheral region of the membrane, whereas integral proteins will be embedded, partially or fully embedded in the membrane. Now, fluid mosaic model. So, plasma membrane. So, it was suggested by two scientists, Singer and Nicholson, that the plasma membrane is having a fluid mosaic model. You know what is meant by mosaic. Earlier, the tiling used to be done of mosaic. We used to say mosaic tiling because chips of different, what to say, stone particles or chips of different particles will be present in this mosaic tiling rather than a uniform color or uniform pattern. Chips you can find, I think even in some of the hospitals still you can see this mosaic tiling. Old buildings also you'll be able to see. Have you seen it before, mosaic tiling?
Yes, ma'am.
Yes, so it is a pattern where different components are used, different substances are used. So fluid mosaic model, it is also known as mosaic model because you can see so many. It consists of different components. It's a mixture of different components because there are proteins, then carbohydrates, then lipids. So different components, a mixture of different components. So you call it as mosaic model. Now fluidity, it is fluidity, the ability of this membrane to move. You know, it's a flexible membrane. It is because of its fluidity, or it's because of its ability to move, that like organisms like amoeba, they can engulf in food particles, so they can carry out endocytosis or phagocytosis with the help of the membrane. Cellulose or cell wall is a rigid membrane, but plasma membrane is a flexible membrane. Shows that the quasi-fluid nature. Quasi-fluid means semi-fluid, semi-solid, means partially fluid and partially solid, or you can say semi-fluid nature of lipids enables lateral movement of proteins within the bilayer. So lipids, it's having a quasi-fluid or a semi-fluid nature, and the proteins embedded, proteins, both integral and peripheral proteins present in this lipid layer can move. They are capable of moving laterally within this membrane. That is why you call it as a fluid mosaic model. Mosaic in the sense that it consists of different components, and these components are able to move within the membrane. And this method or this model was suggested by Singer and Nicholson. Fluidity is a measure of the ability of the membrane to allow movement within itself. So if it is capable of promoting movement within it, then you can call it as a fluid membrane, or it's a measure of its ability to permit movement. Fluidity. If you say that this membrane is having fluidity, or this is a fluid membrane, means it's capable of, or it promotes movement within itself. It's not rigid, just like in the case. That is why children, in plant cells, in pictures also you might have seen comparison of plant and animal cells. So when animal cell is drawn, it won't be having any definite shape, whereas when it comes to plant cell, you know, proper shape. That is because of the presence of cell wall outside the plasma membrane, which is very rigid. It gives it a definite shape, whereas when you compare with it, animal cell, it's a flexible membrane, so you cannot give a rigid shape just like that of a plant cell, because in plant cell they have plasma membrane, but external to that there is one more layer which is known as cell wall, which is a rigid layer. That is a reason for a fixed shape or a rigid nature of the plant cell. But here it is a quasi-fluid nature of the components present, or else you can say it's having a fluid mosaic model, and this model was suggested by Singer and Nicholson. Fluid membrane or the fluid nature of the membrane is important for the cell division. If cell has to divide, the cell needs to grow, and if it has to form intercellular junction, so in between the cells, if junction, some interconnection should be there. You have already learned that there arises some intercellular junction which help in the transport of materials or helps the cytoplasm to interact with each other or to promote the transport of substances across, or secretion. Then endocytosis, I already mentioned. Endocytosis means engulfing something from external environment. For all that, if the membrane is rigid, it's not possible. So it's because of the fluid nature or the flexibility of the membrane that all these functions can be carried out. Once again, fluid mosaic model was suggested by scientists Singer and Nicholson. So according to them, the quasi-fluid nature of the lipids will enable the lateral movement of the different proteins present within this bilipid layer. And fluidity means it's a measure of the membrane to promote movement within itself. And this fluidity or the fluidity of the cell membrane has got several functions. It will facilitate the cell to divide, it to grow, and also promotes the formation of intercellular junctions, helps in secretion and also in endocytosis. An organism which carries out endocytosis, example is amoeba. It can engulf in, when it encounters a food particle, the plasma membrane or the pseudopodia will be put forth, which will engulf the food particle, forms a food vacuole, and it will be taken into the body where the substances or the food present in the vacuole will get digested, and later the undigested matter will be thrown out. All these are possible because of the flexible nature of plasma membrane. If plasma membrane is rigid, it won't be able to, or these cells cannot carry out the following functions that we already listed out: cell division, cell growth, formation of intercellular junctions, secretion, and endocytosis. Okay, any slide would you like to see again? Anything would you like to see again? And then before, yes? Can you repeat?
Not previous slide.
Previous one. Or the last one? This one. Deals with what? Fluid mosaic model? Hello, somebody respond. This one. Previous.
The integral and peripheral road right.
Who is speaking? Buddha, yes, tell me now. What you want to see again? You message me then. I was not able to understand what you were saying. Anybody could make out? No. Okay. Buddha, just message me.
Ma'am, integral and peripheral.
Okay. I'll show it now. Integral and peripheral. Okay. Buddha, this one. The ratio of proteins and lipids differs in different cells. In, for example, in human RBC, 52% of protein and 40% lipids, and the proteins present in the cell membrane can be classified into two types depending upon their ease of extraction or depending upon how closely they are associated with the cell membrane. Integral and peripheral, easy to remember. Integral, integrated into the membrane, fully or partially buried within the membrane, and if they are lying or if they are just attached to the surface, then you can call it as peripheral proteins. These are the two types of proteins. So along with proteins, Even carbohydrates are also scattered within the bilipid layer, forming the cell membrane. Fluid mosaic model is important. Everything is important. There is nothing which you can skip. Okay. So can we quickly summarize what we learned? We'll start with eukaryotic cells. What are the points that we learned about eukaryotic cells? Last period, I could not ask any questions because we were running short of time. Devika, you may start. Children, I used to tell you, don't turn your videos off like this. Yes,
Ma'am,
Devika.
eukaryotic cells consists of protists, animalia, fungi, and
Okay.
plantae.
Yes. Shabiri?
Then
Only one point. Shabiri?
The nucleus has a nuclear membrane, and the cell membrane is composed of lipids and proteins, and it varies from one organism to another.
Okay, along with the nuclear membrane covering the nucleus, what else they have? Something which is membrane bound.
chromosomes.
That is there. What I asked is membrane bound. Certain other structures also they have other than nucleus. Scattered in the cytoplasm, they have. What will be present in the cytoplasm of a cell?
Organelles.
Membrane bound organelles are present. Meherin?
They have complex locomotory organelles and cytoskeleton.
Skeletal structures. Okay. Sarah?
The plant cells differ from animal cells. The plant cells have large central vacuole cell wall, and in animals, what is that? Animals have
Centrioles, which are absent in
animals, plants.
what is the function of these centrioles, Malavika? Which process they help in?
Cell division.
They help in cell division. Alentina, another point. Yes. Alentina, you're checking? Okay.
Which eukaryotic cells are finished?
Where were you? Eukaryotic cells. Where did you go in between? Flying without a ticket?
Ma'am, points under eukaryotic cells are complete.
Anything. Any point that we learned about eukaryotic cells, you can say.
Cell membranes are made up of proteins and lipids.
That Shabari said. All points we covered. Just now we learned a few points, no? We started with the plasma membrane.
Based on the extraction plasma, it can be divided into integral and peripheral.
Proteins. Okay, how are they different, integral and peripheral proteins? Analysts. In a class, 50% of the students are from areas where no connectivity is there, no? Very strange. How many times you will make me say, children? My 50% of energy is getting wasted like this. Why are you so adamant? Yes, analysts. Analysts.
Yes, ma'am. Ma'am, integral and peripheral.
Integral and peripheral.
Integral is the
partially or let them speak, no problem. Partially or
1 minute.
We are running short of time, child. Let them speak, I told no.
It is buried in the partially
buried, partially.
In the
Where somewhere else.
Membrane.
Plasma membrane. Okay. Emmeline, integral, peripheral.
proteins are buried in peripheral parts of
Okay. They are attached to the surface of the
the membrane.
why you're